I'm Erica Ollmann Saphire. I'm a professor and the president of the Jolla Institute for Immunology, and I'm very pleased today to show you the results of the Coronavirus Immunotherapeutic Consortium. COVIC was a consortium launched by complementary funding of the Gates Foundation, NIH, and GHR. And the goal of this consortium at the outset of the pandemic was to compare the hundreds of competing therapeutic antibodies against SARS-CoV-2 to figure out which were best, what criteria you would use to define best, and how you could make cocktails for effective therapeutics. And rapidly, an additional goal became to understand which antibodies would remain effective against the different emerging variants and why. The consortium now includes over 400 different candidate antibodies that are all sent to us by different laboratories and companies that discovered them. And the interesting thing about this study, unlike some of the other papers in literature, is the variety of sources and discovery and selection methods that led to the pool for analysis. Some of the antibodies we're looking at came from survivors of SARS-CoV-1, others from survivors or vaccinees of SARS-CoV-2, as well as nanobodies and engineered multivalent molecules. Another interesting feature of the consortium is that to make the analysis fair, everything becomes blinded. This is what it looks like to us. Every sample has a code name: COVIC-5, COVIC-121, COVIC-173. The contributors are about 2/3 industry and about 1/3, or the remainder, academic and government or nonprofit efforts. What happens is all the antibodies are donated to us, and they are quality controlled and blinded and given a code name. Now, the contributor of the antibody knows which ones are theirs, and our funders do, and my program manager does, in order to communicate with different companies. But I, as the PI, do not, and the database is also blinded. And that's to keep the study fair, so you can immediately download that data yourself and use it for whatever purposes you want to. We receive these antibodies, we codename them, then we aliquot and distribute them to many different COVIC partners, each doing a different kind of analysis that together builds data set. So some participating laboratories are doing binding constants, others are measuring competition, others neutralization, others propensity for escape or in vivo function. So the analysis of many different kinds of features about these antibodies is all occurring in parallel. The data is sent back to us and is cataloged in the COVIC database, which is, or can be found here, covic.lgi.org. Now, one of the first things we did in order to grapple with the number of different kinds of antibodies being sent to us was to sort them into competition groups. And it also became the framework for how we might go and select therapeutic cocktails. This was work done by Dan Bettinger and Partera. He used his LSA instrument for high throughput surface plasma resonance to do fine competition mapping to separate things into 7 major communities against the receptor binding domain, other epitopes outside the receptor binding domain. And then he can also make subgroups within those groups. So this is, for example, a dendrogram of the different kinds of divisions of the antibodies. So on the receptor binding site, so here's spike, it's receptor binding domain, it can lift up. There is a receptor binding motif, that's the part that interacts with the ACE2 receptor. There's an interface and an outer face. Against the receptor binding motif, we can divide it into 3 major sites, and we can subdivide those. For example, site 2 has a lot of therapeutic candidates. We can subdivide that to A, B, and now C and D. So we can come up with fine mapping of the degree of competition in order to build the landscape of this thing. And the binning into the different competition subgroups turned out to be much more predictive than I ever would have thought about the behavior of the antibody. And the behavior could be whether it would neutralize different variants, could be predicted from its competition group, how you would build a cocktail, that we did expect. But also different behaviors like how the IgG would recognize the spike with different stoichiometries. So let me show you what that looks like. So here is a cryo-EM structure of spike. It's upright with 1 RBD standing up. I'm going to rotate the molecule. So now you're standing on the target cell looking down into spike. And there's that 1 RBD. And the colored spots are locations of some of the original mutations that emerged in the original variants of concern, just as anchor points. Chris Barnes in Pamela Bjorkman's lab early in the pandemic defined 4 major epitope classes, like a clock face, 1, 2, 3, 4. Because we have 400 antibodies here, we can come up with finer mapping. And we've subdivided class 1 into 3 different groups, RBD 1, 2, and 3, receptor binding domain 1, 2, and 3. And class 2 becomes RBD 4, class 3 becomes RBD 5, and we've subdivided class 4 into 6 and 7 because they have very different functional behavior. And then within those, we have the finer subgroupings. So in the structural level, this is what it looks like. So I'm showing you a couple of things here. Where it says inner, top, and outer, those are 3 different views of a single receptor binding domain, the inner face, the top face, and the outer face. Where you see a black dotted line, that is the footprint of the ACE2 receptor. Where you see a colored patch, that's the footprint of the antibody. So you can gauge the degree of overlap of the antibody with the receptor. Over on the right are our negative stain EM reconstructions of the antibodies in complex with spike. So I'm showing you a couple of examples here. In red is COVID 259, which belongs to RBD1. And all the RBD1s tend to have the same, they do have the same footprint and similar approach angles. So you can see that 1, the red patch, overlaps that dotted line nearly precisely. These are near perfect overlaps of the ACE2 receptor binding site. Group 2 in orange is shifted a little bit this way. Group 3 in yellow is shifted a little bit that way. 4, 5, 6, and 7 are shifted further outside the ACE2 footprint itself. So look at turquoise and blue and purple. They're outside that dotted line. These are ones that tend to be resistant to the different variants because there are a few mutations in their epitopes. Using that Cartera analysis, we were able to build a complete competition grid across the whole study, the whole COVID study, where antibodies that are dark blue compete, antibodies that are light blue do not compete. And that gave us a framework for picking cocktails. Now, to get some sense of that, let me show you 50 example antibodies. We chose these 50 example antibodies to come from a variety of contributors and a variety of different antibody germlines. We can use this competition grid to make cocktail choices like this. Let's say you had a 2A, and the 2As have some of the most potent neutralization, and you wanted to find different molecules to pair with it. You could pick a 4 or a 5 or a 7, but not a 3 or a 6. This grid analysis and the fine epitope mapping also helped us peg the likelihood to which each of these antibodies would fall prey to one of the different emerging mutations. Those in the orange 2A tend to be sensitive to mutations of position 417. Those in 2B tend to be sensitive to mutations of position 484, for example. But if you look at the bottom right, in turquoise, blue, and purple, groups 5, 6, and 7 tend to be variant resistant, and they always neutralize no matter what new variant emerges. So that was competition. Let me show you neutralization, the ability of these antibodies to block propagated viral replication in cell culture. Now, we come up with a number of example antibodies here too, to give you a sense of how to display the massive data that's in the database. So we've chosen these examples to come from a variety of contributors, a variety of antibody germlines. And we're giving you antibodies across all the 7 epitope groups. So within group 1, you have sample antibodies 69, 259, and 186. And we're going to look at their ability to neutralize a number of different point mutations alone, which we did with the idea that variants might add or subtract a point mutation over time, and also assembled variants. Now, this first slide I'm going to show you was alpha to epsilon plus 2 different versions of immersion MINC, and I'll show you at Omicron, the end of the talk. And then I'm going to distill down all the neutralization data by its fold change. If the mutation or variant wound up being neutralized better by the antibody, it's going to be colored turquoise. If it lost neutralization by the antibody, it's going to be orange. Complete loss is brown. White, no change. So you can use color to assess qualitatively what's going on here. Antibody got better in green, antibody got worse in brown. So there's one molecule in here that's interesting. It's not a natural human antibody, but instead it's 2 copies of ACE2 stuck in an Fc. It's kind of an interesting internal control because you can see it gets better the different variants that emerge. Now, it's still not as potent as a natural human antibody, but it's interesting to see what it's doing. Okay, so we see that overall, most of the 2s and the 4s lose neutralization with the beta and gamma variants of concern. And that is due to these point mutations here, 417 and 384. Whereas those groups 5, 6, and 7 are more variant resistant. Now, of course, there are exceptions. So, for example, antibody 94 somehow has gotten better. Antibody 268 doesn't seem to change. So antibodies are quite individual. There's always exceptions. But overall, we find that most antibodies against those epitope groups are lost by neutralization of emergence of those variants, lose neutralization of those emerging variants of concern, whereas those in groups 5, 6, and 7 seem to do just fine. We get robust neutralization no matter the mutation of the variant at those epitope groups. Another thing we learned by looking at so many antibodies is that it matters how the IgG recognizes spike, not just the FAB fragment. And I bring that up because it's a different way of thinking about it. As structural biologists, we've always thought to look at the FAB, or most of the pictures that you have been shown in textbooks and in papers are structures of FAB fragments bound to antigen. And so if you isolate monomeric FAB fragments, you can easily get the spike fully saturated, the spike trimer in complex with 3 FAB fragments. And so that those images form our expectation for how biology works. But the biologically relevant molecule is a bivalent IgG, 2 Fabs. And so we wanted to understand how that affected binding stoichiometry and occupancy and activity. Now, the first thing to note before I show you that is that we've also tend to picture spike as always vertically upright, because that's how we draw it in structure papers, but it's not. And this tomographic molecule shows that on the surface of the virus, there can be a little bit of rotation, even up to 40 degrees there. So here's what we see. On the left in the gray images are the 2D reconstructions of spike bound to different IgGs. You see some little ghost-like white images. Next over in the blobby cartoon is our reconstruction from the negative stain of the antibody spike complex. And further over to the right is a super simple cartoon that really shows you what's going on. Group RBD1 in red tends to bind monovalently, 3 IgGs to spike. And those IgGs bind 1 Fab on and 1 Fab just out in solvent. RBDs 2, and these are most of the most abundant class of therapeutic candidates, the orange ones, do something different. They bind bivalently. 1 IgG per spike, like goalposts, with 2 Fabs down, and the FC presented up, and sometimes the 3rd RBD left unoccupied. I also wanted to show you the turquoise group, RBD5, because they do something really kind of cool. Many of them cross-link spikes together. So you see here an image of 2 spikes facing each other, and then the IgGs cross-linking them with 1 Fab binding 1 spike, 1 Fab binding another. Now, this kind of arrangement probably wouldn't happen in biology. It happens when you have soluble ectodomains allowed to tumble around. But what would happen in biology is the one over on the right, where you have 2 spikes that are tilted toward each other, and then the IgG binding like this, with 1 Fab into 1 spike, 1 Fab into another spike. And that's a pretty characteristic thing of RBD5. What we actually see are those molecules within 5 that cross-link neutralize. If they don't cross-link, they don't neutralize. And so they're neutralizing really potently, even though they're not at all blocking ACE2 binding. So it's something about how they're cross-linking those antibodies together, maybe blocking fusion, and it can lead to really good protection. So while the group 1s lead to direct ACE2 competitions, we tend to see that the group 2s, RBD2s, tend to bind bivalently within the same spike. Group 5 and turquoise tend to be interspike cross-linking. So we see different characteristic behavior of how the approach angles and geometry allowed by the IgG, depending on whether its geometric footprint is on the top of the spike, shifted. What its position is shows you what the IgG is allowed to do. Okay, so that was a lot of in vitro neutralization in structural biology. What about in vivo protection? Jordi Trillis and Ludus Martinez did for us a lot of mouse protection studies in the K18 mouse model at Texas Biomed. And they began with a prophylactic study. And I'm going to show you a lot of data, again, mapped by color, where the epitope group of the antibodies, so the 7 groups against receptor binding domain, things that bind the N-terminal domain, other RBD antibodies not determined, antibodies that bind the whole trimer, S2, whatever, are along the bottom. Neutralization potency is on the y-axis, where the most potent antibodies are the spots at the bottom, and the worst antibodies are the spots at the top. And the degree to which they could protect the mouse model is given by the color of the spot. So those that gave complete protection, 100% protection, are blue. Those that gave the worst protection, 0%, are brown. Colors in between are colors in between. So you'd like to see blue spots. Where do you see blue spots? Well, you see a lot of blue spots in epitopes group 4 and 5. These tend to be more likely to be protective than, for example, look over in group 2. There's lots and lots of therapeutic candidates proposed in group 2 because you get such potent in vitro neutralization, but they don't always protect in vivo. Now, there are exceptions. So here's a rock star standout in group 1. Here are a couple winners over against the NTD, or unmapped one against the RBD. But here's what we learned. So far, and in general, an antibody must neutralize to be protective. But neutralization alone is not a guarantee. In this brown box are plenty of exceptionally potent neutralizing antibodies that offered no protection in this mouse model. While we were doing that body of work, another variant emerged. So there was Alpha, there was Delta, and then there was Omicron and all the different types and flavors of Omicron. There were so many kinds of Omicron that became a challenge to do this much neutralization of all the different variants on a couple hundred different antibodies. And so we started doing this by blocks, big neutralization blocks. So we initially rapidly screened, just looking at a high concentration and a low concentration of each antibody, and neutralization of each of recombinant VSV pseudovirus of each variant of concern. So for example, this is sample number 96 at a low concentration, and where it neutralizes one of the variants, we have a blue spot, and where it fails to neutralize at all, it's a light yellow spot. So if we now look at the first 397 antibodies we have in 7 different variants, and we look for where do we see blue spots, meaning things that retain neutralization, we tend to see it here. Now, before Omicron emerged, we thought these are the places where we could more reliably get robust neutralization. Some of these different sites, a lot of RBD7, some groups in 5, a chunk at RBD2. After Omicron, and now we're adding 3 more lines to each of these block neutralization graphs, you see a lot of places in the pink boxes that don't have blue spots below them. A lot of antibodies failed to neutralize Omicron. So now who do we have left that robustly neutralizes all the different types of Omicron, as well as the other variants? So these are the antibodies we'd want to consider going forward. And those are now in the pink boxes. And whereas 23% of the COVID panel could retain neutralization against G614 and beta and delta and mu, only 5% neutralize all of them but Omicron, or all of them and Omicron. But that's still pretty good. There's still a number of antibodies in here that neutralize all of the known variants plus Omicron. And we learned something interesting here about the geometry. So let's go back to RBD2. There are more antibodies submitted to COVID as therapeutic candidates in RBD2 than any other class. And RBD2 are the antibodies that are more likely to bind bivalently, 1 IgG per spike with the 2 FAB arms on 2 adjacent RBDs, and the third one left empty. Now, an IgG can bind spike a couple of different ways. It can bind monovalently, and that's 1 Fab of each IgG on. So 1 Fab on, 1 off, and you might have 1, 2, or 3 IgGs bound per spike trimer. An IgG can bind bivalently, and that's goalposts with both Fabs of each IgG simultaneously anchored. That's only 1 IgG allowed. Or it could be bivalently plus an extra antibody. So we've got the goalpost binders on copies 1 and 2 of the trimer. The third copy of the trimer that's open could be bound by another IgG. So we have 1 anchored down like goalpost bivalently and an extra IgG bound to that third RBD with its FAB free. Or it could be incompletely bivalent. Some copies viewed in the electron micrograph might be bound by bivalently, and some might be bound by one. So it could be a complete bivalent binder, or a bivalent plus a spare antibody, or partially bivalent. We can characterize all that by electron microscopy. So we can look at each one of the antibodies in the consortium. So for example, we're looking at sample 294, sample 299, sample 360. And we can look at all the different examples of all the different copies of a 294 spike complex. And we can come up with what percentage they are. Each one of those copies of the complex are bivalent, monovalent, or whatever. We find a couple of things. Of those antibodies in group RBD2 that retain neutralization of Omicron, they all bind bivalently. So they're either like at right half, just bivalent. You see those two orange bunny ears. Those are the two FABs coming down. The FC would be up above. It's disordered. You can't see it, but it's one Ig per spike. Or if you look at the example of 3 on the left, it's bivalent plus a spare. Those are two FABs from a single IgG, and the third one from another IgG. So if they retain neutralization of Omicron, they tend to be bivalent. Of those in group RBD2 that do not neutralize Omicron, now this is interesting because they bind essentially the same footprint. They tend to not be bivalent. So if you look at the two on the left, they bind monovalently, one Fab on, another one hanging off. If you look at the ones in the center, they are partially bivalent. So like half the copies are bound bivalently, half are not, or sometimes 75-25. So among those so we have a lot of antibodies against the same footprint. Then here, 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 and here. But by binding that spot, some do it bivalently, some monovalently, some do not. So antibodies 49 and 73, for example, bind monovalently. The ones in the middle, antibodies 147, 43, 45, and 90, bind 22 to 50% monovalently. So there's some percentage that's monovalent, and there's one guy that's bivalent. These guys, by binding bivalently, tend to retain neutralization of the variant with so many different mutations. So something about being able to hang on through avidity helps retain the binding despite mutations. And whether or not this antibody can bind bivalently has something to do with its approach angle. If it comes from the side, there's no way it's going to be bivalently. It's got to be a position at the top. And something about its recognition geometry. And something about the being able to retain that avidity confers this robustness, or can maintain binding despite all the different mutations. And you can see this in the neutralization. So if you look at whether each of these IgGs, their ability to neutralize D614G in navy or Omicron BA1.1 in light blue, you can see antibodies like over on the left, 361 or 290, that neutralize the variant and the original about the same. The one in the middle suffers for the variant, but 4 of these as an IgG, seem to neutralize the Omicron variant pretty well. And you can see the importance of avidity when you cleave these IgGs down to FAB fragments. So if you look at the antibody over on the left, 361, as an IgG, he didn't seem to care whether he was looking at the original virus or Omicron. But as the FAB, if you compare the brick line to the salmon colored line, he really can't neutralize as a Fab. He can only neutralize the original viruses in FAB. So being able to hang on avidly helps improve the neutralization. Now, all of that data is available in the COVID-DB Consortium website. And a study of the scale would not have been possible without so many people taking a bit of it. I'd really like to thank our funders, the Gates Foundation, NIH, and the GHR Foundation, the Overton family, for supporting the study. I'd like to thank my program manager, Sharon Schindel, for coordinating all the samples in and samples out. Catherine Hasey for doing so much neutralization, Hao-Yang Lee and Heather Calloway for spearheading the structural biology. Dan Bettiger at Cartera led the epitope competition mapping. Georgia Tamaris, binding constants. Bjorn Peters' lab developed the database. Others at other institutes did other kinds of neutralization, Fc effector activities. I'd like to particularly thank Texas Biomed for so much in vivo work. I'd like to thank all the COVID participants for contributing their samples and their analysis so that we could build this lasting database of information for continued years of this coronavirus pandemic and other coronaviruses to come. It's good to understand the landscape of antibody activities, what is protected and which are lost. Because at the monoclonal level, we can get the information we need to understand what may be happening also at the polyclonal level. And we can use this information to engineer new antigens to make longer lasting vaccines. And thank you for the opportunity to speak.